Hello, thank you for tuning in today. I am going to tell you about my work neuroimaging in young children, and specifically in a cohort of young children from the APrON study. So early childhood is a period of rapid brain development, as well as cognitive and behavioral maturation. And it's also a time period when many cognitive and behavioral problems, such as those associated with neurodevelopmental disorders, first become apparent. And while we know that brain development is happening during this time, we actually don't know very much about how that brain development proceeds and how it varies regionally and how it's related to these emerging cognitive and behavioral skills. And this is because previous neuroimaging studies have largely focused on infants or children school-aged and older. And that's largely for practical reasons, because young kids tend to be squirmy and somewhat difficult to convince to lie still in an MRI scanner for an extended period of time. And on top of that, MRI is a noisy and claustrophobic environment, and it's unfamiliar for most kids. So they can just be challenging to get good brain data on. But this is something that we've been working really hard at the University of Calgary and the Alberta Children's Hospital to try to get high quality brain data in these young kids so we can really look in detail at how brain development is proceeding across this age range and how it's related to these cognitive and behavioral skills. So at the Children's Hospital, we have an MRI scanner, a research dedicated MRI scanner, and we've put a rocket ship facade on it. So it looks like a rocket ship, and we can tell kids that they're going for a ride in the rocket ship. We also have a mock MRI scanner, and this is a place where they can practice. So they can put their parent in, they can put a stuffed animal in, or a research assistant, and practice without the time constraints of the real MRI scanner. And this mock MRI scanner looks and feels and sounds just like the real thing. We've also constructed an astronaut training program that goes along with this. So level 1 is completing the mock scan, and level 2 is completing the real scan. So kids can feel like they're advancing through the levels. And then finally, we have a children's book about this process. It's about a child getting invited to come participate in a research study, coming to the Children's Hospital and going for a ride in the rocket ship. And I've put the link at the bottom here in case anyone's interested in taking a look at it or potentially even using it in your own research. So we've had a lot of fun using these techniques with kids, and we've had some great success. And I think a lot of the kids have had fun with the process too. This is just an example of a really sweet letter and picture that we got from one of our participants. So as I said, we've had pretty good success using these techniques to get data in kids. And thanks to really amazing efforts by my research assistants and my team, we have collected a data set that's now 562 scans on just over 200 typically developing children. And most of these children were recruited from the APrON study. So kids were recruited between the ages of about 2 and 5. And then we've been following them semi-annually or annually thereafter. And so the oldest scans in our data set right now are about 8 years old. These kids were scanned on a research dedicated MRI scanner at the Alberta Children's Hospital. And we get a number of different types of imaging, including diffusion MRI, anatomical MRI, and passive viewing functional MRI. And I'm going to tell you in a second a little bit about what those different techniques can tell us. And then at the same time, we get a very short battery of language assessments on the kids, particularly related to pre-reading skills. So one of the techniques we get is diffusion MRI, and this is really sensitive to water motion within the brain. And because the brain is ordered, particularly in the white matter, we get ordered diffusion. So water is more likely to diffuse along the length of an axon in the white matter than it is to go across that axon. And this is because of the axon membrane as well as the myelin sheath that surrounds it, both of which restrict water movement. So water tends to follow this path of least resistance, and we can measure that with MRI, and then we can extract some parameters that are very useful for characterizing the microstructure of the tissue and can tell us something about the packing of these axons, the amount of myelin, and sort of how ordered those axons are. The primary measures that we tend to use with diffusion imaging are fractional anisotropy, or FA, and mean diffusivity, or MD. So FA is a measure of the directionality of diffusion. So the more ordered and the more directional diffusion is, the higher the FA value. So if this is a sort of diffusion pattern, the higher diffusion in one direction is compared to the others, the higher the FA value. And this is an FA map of the brain, and the high values here, the bright spots, represent high FA, or very highly ordered tissue, and this coincides with white matter tracts within the brain. Mean diffusivity is a measure of overall water movement within the brain, and it tends to be high in areas with very few restrictions to diffusion. So the cerebrospinal fluid, for example, has very high values of mean diffusivity because water can move around very freely, whereas any of the tissue areas, the white matter and the gray matter, tend to have lower values of MD because diffusion is more restricted. So this would be a representation of a lower MD value versus a higher MD value. We can also get some basic directional information about water movement within the brain. And this is an FA map here, but it's color-coded by the primary diffusion direction, with red representing left-right, green front-back, and blue up and down. So you can now tell not only what's white matter, but which direction those tracts are ordered, which can tell us a lot about how the brain is organized and how that structure looks. And we can also sort of play a fancy version of connect the dots, which lets us actually map out the white matter tracts within the brain and get something that sort of looks like this. So we can actually take each of these individual tracts within the brain and query them to measure FA or MD, for example, and get a picture of how connected the brain is, to look at how that relates to cognition or behavior or environment or age. Now we can also look at brain function, and we tend to use a technique called resting state FMRI to use this, or at least this is what we use in our study. And this looks at the blood oxygenation level dependent signal. So sort of the brain activity during rest, or in our case, the kids are actually watching a movie, but they're not doing a specific task like reading or finger tapping. So we look at the fluctuations of brain signal within the brain multiple times over a period of several minutes. And by looking at that signal over time, we can get an idea of how functionally connected the brain is, or how synchronous the activity is. So we look at one area, for example, and we take the time course of signal there, and we correlate it with every other area in the brain. And then if those two time courses are very highly correlated, we would call those brain regions functionally connected. In some cases, we get negative correlations, and we would call those anti-correlated or negatively connected. We can also look at specific brain networks. For example, the default mode network here, where we take known brain regions that are part of a specific network, and we look at how well connected that network is. And then again, we could compare that with age, cognition, behavior, any number of variables that we're interested in looking at. So my group has been using these techniques to look at brain development in kids. And again, in that sample that I told you that was drawn from some of the APrON sample. And so what we've seen is really nice, clear brain development happening in the white matter. So this is whole brain white matter. We see increases of fractional anisotropy and decreases of mean diffusivity. So really indicating that there's substantial development going on during this age range, which we expected, but also showing some nonlinearity. So faster development at the early age ranges, and then sort of tapering more towards the end. But the interesting piece of this, I think, comes when we start to look at regional variation within the brain. So we did that. We looked at a number of different white matter tracts across the brain, and these fell into three sort of main categories. The first category of development were tracts that showed slow development, relatively speaking, but had very high values of FA at the start. So these are regions like the corpus callosum in red behind there and the pyramidal tracts in green. And this suggests that these tracts have actually undergone quite a bit of development before the start of our age range and are continuing, but they're sort of in the later phases of their development. And then we had another group of tracts. These are frontal temporal brain connections that we know are involved in higher cognition and emotion processing, for example. And we see, again, relatively slow development, but much lower initial values. So 0.36 here compared to 0.52 here, indicating that these tracts are not very far along in their development. In fact, these are more of the slow and steady tracts that we actually know will continue to develop into the 20s. And then finally, we saw this group of association tracts that had very substantial development across the age range. So you can tell that these are the tracts that are really undergoing a major change in this early childhood age range. And these tracts are involved in a number of different processes, but among them is language. And so it sort of makes sense that a lot of these functions that are improving in early childhood, the underlying neurobiology is also changing rapidly. Now, one of the things I want to point out as well when we look at these figures is that there's actually quite a bit of variation across individuals. So each little gray dot here is one scan from one person, and each line is that person's best fit trajectory across their ages. So there's huge variation among 3-year-olds here, for example, almost as much variation as there is between the average sort of 3-year-old and the average 7-year-old. So major variation. But when we zoom in on individual participants, we actually see that they tend to stay along the same curve. So those that are above the curve tend to stay above, and those that are below tend to stay below. There's a little bit of variation, of course, over time, but fundamentally, most of the kids are following a similar trajectory. And I think what this really highlights is the importance of this longitudinal imaging, because it may not be quite as much about where somebody starts, but rather whether they're continuing along that same trajectory or not. So now I want to talk a little bit about how some of this brain development is potentially tied to language and reading abilities. So reading is obviously a really important skill in our world. It's very critical for success. For example, it's associated with academic performance, with mental health, and with career prospects. And a lot of the early reading skills emerge in early childhood. In particular, phonological processing and rapid naming skills in early childhood are strongly predictive of later reading ability. So those are two of the measures that we looked at in our sample. Now, in terms of brain structure, the reading network and brain function, the reading network is left lateralized for the most part and really relies heavily on areas in the inferior frontal lobe, the temporal parietal lobe, and the temporal occipital area. And better structural connectivity among these areas is associated with better reading skills in older children and in adults. And in particular, we've got the ARCUATE fasciculus, which is a tract connecting the frontal and temporal areas through some parietal areas. And it's really instrumental in a lot of the reading and language processes. In terms of its volume, it's highly leftwardly asymmetric in the vast majority of children, adolescents, and adults. So about 90% of them display leftward asymmetry of this tract. It's also functionally asymmetrical, again to the left side, which is present in infants, but also shows increases from childhood to adulthood. So we were really interested in how this asymmetry perhaps emerges in early childhood and how that supports reading or early language abilities. So we looked at structural and functional asymmetry of this area. And what we saw for sort of macrostructural asymmetry, so this is the number of streamlines in the tract in each side, essentially getting at volume or size of this tract. We saw that most kids were leftward lateralized. Anything positive here is leftward asymmetry, and anything negative is rightward asymmetry. So most kids are leftwardly asymmetric. And furthermore, this doesn't change with age. So most of the kids, even the very young kids, are already displaying this asymmetry. FA showed a similar pattern where most of the kids are leftwardly asymmetric, and this is constant across age. So the asymmetry of the FA values isn't changing substantially. But when we looked at MD, we did see decreases of this asymmetry with age. Now, because MD actually decreases with age itself, this decreasing line indicates more leftward asymmetry in the older kids than in the younger kids. And then when we looked at function, we saw, again, increasing leftward asymmetry, both in terms of intrahemispheric and in terms of interhemispheric functional asymmetry. So we've got this macrostructural asymmetry that's established early. It's already present at age 2. But then we've got this increasing microstructural asymmetry and increasing functional asymmetry with age, really suggesting that the brain is sort of refining itself, and perhaps this is related to the ongoing or the emerging reading and language skills. So that's the next thing we wanted to look at. Now, when we look at FA values themselves, so not asymmetry, but just FA values in the left hemisphere, we see a very nice relationship with the phonological processing skills. So more connectivity of that tract, higher FA, is related to better phonological processing skills. So better language, better brain connectivity. But then when we looked at asymmetry of any of our measures, so the number of streamlines, the FA and MD values, and the functional asymmetry, none of them were significantly correlated with our language measures, neither phonological processing nor speeded naming. Which is interesting because we expected to see these to be related, but perhaps it indicates that this relationship really is emerging and isn't quite present yet in these young kids. So I think it's going to be important for us to follow these kids later and really see what happens and whether we can detect those relationships at older ages. Now, finally, I want to give you just a quick example of how we're using this data in typically developing kids from the APrON cohort to help inform research in an atypical cohort of kids with prenatal alcohol exposure. So prenatal alcohol exposure is associated with a variety of difficulties, including cognitive, behavioral, and neurological problems, and also occasionally with facial dysmorphology. The neurodevelopmental disorder associated with PAE is fetal alcohol spectrum disorder, and this is quite prevalent. It affects about 4% of people in Canada, which is huge. It makes it 2.5 times as common as autism. So PAE is associated with widespread brain alterations, including lower brain volumes, and in older kids, it's associated with lower FA values. But we really don't know what's happening in young kids with prenatal alcohol exposure. And this is really important because this is when those behavioral and cognitive problems often emerge and first become apparent. But we don't know how the brain underlies that. So we looked at this in a group of 49 kids with prenatal alcohol exposure. Most of them had 2 scans. And we compared this to the typically developing group that I've already shown you. So we looked at a number of different white matter tracts, and we measured FA within those white matter tracts. And interestingly, what we saw was higher FA, or that's more ordered brain structure, in the kids with prenatal alcohol exposure compared to controls. So a bit surprising. We don't tend to think of better brain organization as being present in a neurodevelopmental disorder. But in fact, when we dug into this a little bit more, we think what we're seeing is accelerated maturation that perhaps is not optimal. So when we look at developmental trajectories, we see that the children with prenatal alcohol exposure, they start higher here for FA, but they're not changing as much, whereas the controls are having more substantial development here. And it looks like about age 7, they're crossing, and then the controls will continue developing while the prenatal alcohol exposure kids perhaps will not undergo as substantial of a development. So they're perhaps plateauing earlier. And this fits actually with some of the other literature on early adversity, in particular threats or traumatic stressful events in childhood. And so perhaps prenatal alcohol exposure is its own sort of form of early adversity that's associated with accelerated maturation. So for example, early threats are associated with accelerated DNA methylation, as well as advanced puberty. And traumatic stressful events in childhood are associated with a more advanced pattern of brain maturation. So this may be what we are seeing in our young kids with PAE. So now, just to wrap up with a few thoughts, high quality MRI is possible in young kids, and it's really important to understand the neurological correlates of a lot of the behavioral and cognitive problems or maturation that we see. Longitudinal data in particular is really critical for understanding development trajectories and can tell us a lot about who is developing appropriately and perhaps who is not. And then we see really extensive brain development in young kids, as well as substantial regional variation across the brain. And then finally, in children with prenatal alcohol exposure, we see altered development trajectories that suggest perhaps accelerated brain maturation. So finally, I would like to thank my lab. I would like to thank my collaborators and my sources of funding, and of course the APrON study for allowing us to contact these participants, and thank all the participants for giving us their brain data. And finally, I'd like to thank you for your attention today.